Thank you, Hans, for the nice introduction. It's true that I grew up at Sherm, at least by now I can say that. I've been working to develop and apply Intel NMR methods for a few years, and now we are specifically focusing on the real-time aspect of NMR, which is something that among the atomic resolution techniques, I think NMR is one of the leading methodologies to do that. So I'll first show some brief introduction on in-cell NMR, what is it and what we do here since a few years and what we have developed. And then I will focus on the real-time NMR part. So in-cell NMR is an application of high-resolution biomolecular NMR, which allows directly to obtain information at the atomic level, so with very high structural resolution in the physiological environment of a living cell. As such, it's a very unique and powerful way to obtain biologically relevant structural information and therefore functional information in the context of a living cell, and as well as a way to validate any possible data obtained in vitro to assess whether that can reproduce and actually make sense given the cellular context. So this is the method that we have developed at Sherm a few years ago, which is based on the protein overexpression in human cells. So we're focused on studying proteins, and we can overexpress them in HEC293 T cells, which are very useful because they are robust and allow quite high levels of protein expression. So they can reach the levels needed for detection by NMR, which is intrinsically not so sensitive as other techniques such as fluorescence microscopy. So this is only one of several approaches that allow observing proteins in cells. The other ones mainly rely on protein delivery in cells. So one protein is purified outside of the cell and then is delivered with several methods, which has several advantages. This is an alternative approach which relies on the expression inside the cell. And as such, it has a few advantages because there is no handling required for the protein of interest, and it can be also, that's useful for some sensitive proteins which are prone to aggregation if manipulated in vitro or need oxygen. I mean, they're sensitive to oxygen or to other environments. And it's quite robust because protein overexpression usually is performed at high level for different proteins. So by providing isotopes, we can also enrich the protein with carbon-13 or nitrogen-15, and this allows multidimensional NMR experiments on living cells. And in the years, we have applied these methods to several kinds of studies. Mostly we focused on protein folding and maturation and the study of aggregation mediated by the dependent on the pathological mutants of proteins, but we could also extract, purify organelles and do inorganelle NMR. And another important aspect that in-cell NMR is very useful for is the direct observation of the cysteine redox states of proteins. Then we also worked to integrate this technique with imaging techniques. So this is just a quick overview of what we do and we did. Recently, we focused on the possibility to apply this approach to screen small molecules, potential drugs in living cells. So the rationale behind that was that in-cell NMR, being a structural technique able to investigate binding events in cells, it could help the drug development pipeline, which is intrinsically very inefficient and expensive process, between the steps of lead optimization and preclinical assays in which you have found promising candidates in vitro, but then you need to test for their efficacy in tissues, so animal tissues, before going for humans. And there is a very high attrition rate in these steps of the pipeline, where some drugs which were very active and promising in vitro, then they reveal ineffective in the tissues. for several reasons. So we think in-cell NMR is a very good way to approach this kind of problems and screen molecules inside the intact cells. And to do that, we focused on a protein which is both a model system because it's extremely well characterized, carbonic anhydrase 2, and also is a pharmacologically relevant target because its CAs are involved in several pathological states, and there are huge libraries of inhibitors constantly developed for these proteins. So a nice thing about this protein is that it behaves well in terms of interactions within the cell. So we get nice HSQC spectra. We can actually map the interaction between a protein and a drug inside the cell by looking at the perturbation of these the chemical shifts in the spectra. Another interesting fact about this protein, which is a metalloprotein, there is a zinc coordinated by 3-histidine, is that a few proton signals, including the 3 from the histidine ligands, from these zinc ligands, the histidines, fall in a region of the proton NMR spectrum, which is totally free from any other background signals from the cell, which means that even by doing 1D proton NMR without any isotope labeling, we can observe these signals, and we can see whether they are perturbed upon ligand binding or not. So that makes for an excellent model system to do drug screening. So that's what we did in the first place. And by screening, by testing a series of these experimental compounds, plus 2 approved drugs, cetosoliamide and metazolamide, we could easily identify those who could penetrate the cells and bind the target, whereas several others, it was easy to see from the spectra that they didn't bind the protein at all. And by the way, this did not correlate clearly with the Ki. With the same approach, we could then perform a dose response analysis by treating the cells several times with different doses of the same ligand. And then by fitting with different models, we ended up with a diffusion limited model in which the rate determining step is the diffusion of the ligand through the plasma membrane. So we get sort of linear response in time, both in dose and in time. And that was interesting in itself because it could explain some effect, some information that we had on the model compounds which were approved drugs in terms of permeability. Because, well, first we had a few negative compounds which had experienced, which showed a very low predicted in silico permeability, which correlates well with our findings. And then we found an interesting correlation between the permeability, which we calculated based on the dose and time dependent binding, and the recommended dosage of these 2 drugs in the treatment of glaucoma. So we thought that this striking correlation makes a good argument in favor of in-cell NMR for this kind of studies. Now, what's the problem here? Everything I showed up to now was made by treating cells in an incubator, then trypsinizing them and making a pellet inside the NMR tube. So this increases the cell density to a huge amount. So we got roughly 30 million of cells in 150 microliters, which leads, unfortunately, to a very quick cell death, or at least instability of the sample, which then leads to cell death. And that's why we keep these experiments very short, ideally below 2 hours each, which then limits a lot the throughput of the technique and increases the effort required to produce many samples. So what's the solution for that? The solution is to provide a continuous exchange of medium so that new fresh medium is provided to the cells and the byproducts of metabolism are discarded. And this can be realized with a flow bioreactor. This has 2 benefits. One, it allows us to record longer NMR experiments. So multidimensional virtually NMR experiment could be recorded if we had more time to do so. But also, and I think more importantly, we can monitor events in real time. So things that change over time can be monitored in a time-resolved manner. And so in collaboration with Bruker, we modify a flow unit from Bruker, which is the Insight MR. It's a commercially available device, which is simply a flow unit that allows to replace the content of the tube. And we had two alternative designs. So we developed a sort of a modular bioreactor, one that could be adapted for different needs. In the first iteration, we replaced the inlet with a coaxial microdialysis membrane, which allows the diffusion of small molecules through the membrane, so that the flow of nutrients of medium is inside this coaxial membrane, and the cells are suspended in the NMR tube here, in the glass tube, and they are blocked so they cannot leave the magnet. So the cells are kept in suspension here. This approach is probably more useful for suspension cells. Whereas for adherent cells that don't like to stay in suspension, we then opted for a more classical design where the cells were encapsulated in agarose threads, and the fresh medium flows directly to the bottom of the tube. With this latter approach in particular, we could measure very high cell viability for after long experimental times. So in the first design, we keep the cells in suspension, and then the nutrients can diffuse freely. And this was, the cell viability was checked by trypan blue and by phosphorus NMR, in which we could monitor the signal from the beta ATP, which is only present in ATP and not in ADP. So from this signal, you get a clear picture of the metabolic activity of the cells. And that was sufficiently high, even for higher times, like up to 2 days, although then the cell viability started to decrease, probably because of the very high density of cells, and because we use the hex cells, which are adapted for growing as a monolayer and not in suspension. However, compared to the control, which is the red here, you can see that there is a huge difference in terms of glucose concentration in the medium, which in the absence of flow is quickly depleted to almost 0, and the presence of ATP, which also drops to 0 as soon as the flow is stopped. In the second design, the cells were encapsulated in agarose, and this even improved even more the viability of the cells for up to 72 hours. So actually, we could probably go even to longer times with this design. And same control, so we got ATP for 72 hours, ATP production, that's the black dots, where it has absence of flow or just 24 hours, and then shutdown of the flow quickly lead to a decrease in disappearance of the ATP signal. So with this technique, we could perform real-time ligand binding in-cell NMR. That's what we did. So we took back our test compounds, the cethozolamide and methozolamide, which are the inhibitors for carbonic anhydrase 2, and recorded NMR in real time. So that's the 1D spectra. They are all stacked. So in time, we got these nice movies where you can see the signals of the free protein which go down and the bound protein which go up. And this is a second ligand. You can immediately appreciate that here the kinetic is different. And that is, I can anticipate that that's due to the fact that this ligand is more penetrant. It can cross the plasma membrane faster than the first one. So each of these slides of the movie looks like this. That's a bit noisy, but they are about 15 minutes each experiment. And by the way, one can choose to take shorter experiment with the higher noise, but higher temporal resolution, or longer experiments with lower noise and lower temporal resolution. That will affect the outcome of the experiment. But anyways, we then were faced with another purely practical issue: how to get information from this data. We cannot simply manually integrate each of these signals, and then we have overlap in some regions. So we had to find an automated way to do that, quantitative automated analysis. And to do so, we relied on MCR ALS, multivariate curve resolution alternate least square fitting, which is quite straightforward. It's a very simple algorithm that exploits the fact that the dataset matrix, which is B, can be expressed as the product, the matrix product of the concentration matrix. So you have the data, raw data, which is in this case chemical shift time per time. So in this two-dimensional array, which can be expressed as the concentration matrix, which is the number of species, in this case 2 free and bound protein per time, and the transpose matrix of the pure spectra, which is chemical shift per species. So, and then plus, if you compute this, they will also have a residual error matrix. So by iteratively improving, recalculating these two, the C and S matrix, and then imposing a constraint where the fitting stops when E reaches a minimum value, you can reproduce you can extract from the raw data the concentration profiles in time and the pure spectra. And in this system, but in principle, in many other cases, this gives a beautiful result because totally automated, in a totally automated way, and quite easy to implement even on a personal laptop computer, such as the one from which I'm talking. And you get from the raw data, in few seconds, you get the pure spectral components, which, as you can see, are almost noise-free. So it's also cleaned up the data from noise because we had many spectra to use within the algorithm. And the concentration profiles with quite a good quality in terms of noise of the profile. Then by also changing several parameters for the fitting, we ended up with the same curves, basically. So it's also quite robust. And now these concentration profiles can be fitted with kinetic models as one wishes. So in this case, we got a very good fitting with the diffusion limited binding kinetics that we had shown in the previous work and the static NMR work. And they were confirming the difference between these two compounds in terms of permeability of the plasma membrane. So methacrylamide is 10 times faster than methacethylamide. And then this algorithm is quite flexible in terms of the input data. It can also be applied on two-dimensional spectrum. So we repeated the analysis on a set of 2D HSQC spectra. In this case, this is superoxide dismutase protein, so that's a totally different system in which we induced the formation of the internal disulfide bond by treating the cells with an active molecule epsilon. So these where we previously showed that it would induce the formation of the native disulfide bond in the protein. So we just monitored that as it happened in real time, and we got curves also from the 2D spectra. These are the pure components retrieved by the MCR-ALS algorithm. So, and then this is just a simple bi-exponential fitting that has no biological meaning. It's just to show that one can fit the data if you know the model or if you make hypotheses. So this approach is quite robust and versatile and can be applied to 1D NMR, to 2D NMR in principle to higher dimensionality. It would increase the computational requirements a little bit, however. And it's very useful to then study kinetics in cells. And not only that, because then we recently went into more depth in the carbonic anhydrase binding with the model compounds and with experimental compounds, and we assessed whether amino acid type selective labeling was useful. And in fact, it is, because we can, by labeling selectively the histidines coordinating the zinc, we can increase the dimensions along which we have chemical shift perturbation. So you see the perturbation in 2D instead of 1D. You have no more signal overlap, not anymore. So that's even easier both by eye and by the algorithm to separate the pure species than with the 1D spectra, which then translates in the fact that you can then work with more than 2 species. You can observe the sequential binding of different ligands to the same protein. Now, this was kind of a game. I mean, it's not really, it doesn't really make much sense because we knew the affinities of these compounds and we treated the protein with increasing affinity compounds. So we expected to get complete binding for each of them. But that means that you can work with unknown ligands and also potentially mix them and perform competition binding experiments, which means that we could extract the dissociation constants, intracellular dissociation constants by real-time cell NMR. So that's also a thermodynamic information, not only a kinetic one. So I think this gives you an idea of what can be done with the bioreactor. Although I'm focused on the protein-ligand interaction, you realize that these methods, in principle, can be applied to any intracellular event, where anytime you see a signal which responds to your stimulus in a way that you can measure, in principle, you can apply this, the bioreactor to obtain, to study intracellular kinetics. So I hope that was interesting. I want to thank the people who were involved in these studies that I showed, and thank you all for your attention.